Thank you, Yifan, for giving me the opportunity to speak at this interesting meeting on brain-inspired technologies. I have to say that this has been part of a joint collaboration between China and Cuba. And here you see part of my lab in Chengdu at USTC. I have to say that this is also part of a larger collaboration, which is the Cuba-China-Canada project, as well as the Global Brain Consortium, of which I will speak further on. Now, in the Cuba-China-Canada project, our main collaborator from Canada is Professor Alan Evans from McGill University. He is the PI of a $300 million project known as the Healthy Brains for Healthy Lives. And what this is trying to do, what this project is trying to do, is to find biomarkers that can be reduced by nonlinear multivariate analysis methods to a much lower space manifold, which can help describe the normal brain development and abnormal deviations from this development. Here in this slide we can see, for example, the red main trajectory there would be normal development. On the other hand, the green one would be a deviation from normal development. And then perhaps a branch that we see from the green one is a return through treatment towards normality. Now, this whole idea is predicated on the basis that we can find biomarkers that actually will reflect the activity and development of normal and abnormal brain function. Has the basic idea that up to now there has been a failure of psychiatric classification of disorders and even of many neurological disorders, because they assumed a direct relation of genes with brain states. The concept of pleiotropy has come into effect, and that is that the genes control different brain states indirectly through mechanisms that are known as endophenotypes or intermediate processes that can themselves combine. So the relation between gene and let's say these intermediate mechanisms would be simple, but then they would combine, and that would in different ways, and that would give us the difficulty in being able to parse or detect the different types of diseases just from the symptoms. Now, a main candidate to be one of these intermediate mechanisms are the connections of the brain. And here we see from an example of what's known as functional MRI, the creation of a map of connections between different brain regions. This has been well studied, and in fact many people consider that it can be a model for detecting biomarkers in some way that would allow us to do the program I said about characterizing disease progression. Look at it. In fact, we have several position and review papers recently, and this is one that we published in Nature Neuroscience, and the position here, and this is a scheme that I have proposed, is that we actually have a level in which there's connections of neural masses. These neural masses are reflected on the surface of the head. That's what you see on the bottom left of the slide. And then we have a reflection due to this of correlations or association measures that tell us how much sensor activity covaries. To consider that sensor activity, let's say correlations, are in any way a reflection of what's happening at the upper left, which is at the narrow level, is really to ignore the physics of the problem. So we have to perform an inverse problem to find an inverse solution to be able to then try to estimate the unobserved connections between narrow masses. This is what's known as the EEG inverse problem. One of the main challenges we've been facing recently is to try to solve the problem of leakage in source activation and source connectivity. We know that the true narrow sources are never reconstructed perfectly. In fact, if we look at the forward problem, we go from a certain connectivity pattern. We will put the precision matrix as theta. That will generate currents, and that will generate the observed voltage. The inverse problem is going in the opposite direction. And what we want to do is to reconstruct from the voltages the connectivity patterns. Some of you have heard some of my talks and know that one of the problems that exists is known as leakage. That is, if we have an actual connection here in blue, then when one does a reconstruction, there's usually a spillover of both activation and connection. Actually, Deirel, who is there, can show you how even in simulations with a perfect reconstruction of activation, you can have leakage in connectivity. Now, this is more pronounced in what we call two-step connectivity estimation. This is when you do a first step and use a linear method or a nonlinear method to estimate the activity at different cortical voxels, and then subsequently and independently try to estimate the connectivity pattern. One would ideally like to bypass the first step, which is the estimation of the activation, and go directly in a single step to the estimation of the connectivities. This would supposedly eliminate leakage both in activation and in connectivity. What Deirel has come up with, and the group that works with him, is the idea that we can do an iterative estimation of activity, connectivity based on the information that comes from the sensors, and in this way achieve a close approximation, that is to say, to achieve the evidence that would be obtained with, in the Bayesian sense, with a one-step method. Now, this method is what we call BC Vareta. It's been developed in our lab by Deirel, who has been heading the group, by Arioski, who is the software engineer behind all the work, and by Rigel Wang, who has been doing a lot of the calculations and comparisons with other methods. You really have to know that there's a further team, but these are the three main participants. And the basic idea behind BC Vareta, of which there's a toolbox on GitHub, is to try to extract the magneto or electroencephalographic spectral activations and do this through a sparse Hermitian graphical model. That is to say, to use a Gaussian graphical model, but in the frequency domain
So that is why it has to be Hermitian. Uh, Basically, it has two steps. One which does a screening of active voxels, and it's based on a method developed years ago by Deirel, which is the elastic net SSBL source screening, and then what we call the hidden graphical Gaussian model, source model, which is putting up sparse prior on the connectivity matrix. I want to emphasize that this is a prior on the Cartesian product, that is to say, of all pairs of sources, and it is a third level analysis. What happens now is that an arbitrary covariance structure is posited, and the linear methods that are being used, like Loretta or even some of the beamformer methods, and then subsequently, in spite of the, let's say, the misfit or the inappropriateness of this prior, to go ahead and estimate the covariances. This is overcome in this procedure. The details you can get from data. Now, basically, what we did were two sets of work. One was to generate a as realistic as possible from work that we did many decades ago with Roberto Pascual while he was doing his PhD in CINEURO, in which we decomposed the EEG, the resting state EEG, into a 1/f component, which we called C, and then the alpha peak. Now, we used data, this data that we have from normative studies, to generate a simulation where there was a specific connectivity pattern which was quite, let's say, focalized with delays, and you see it here. This is the connectivity matrix. The delays are not shown. It would have to be a tensor. More about this later. And then, actually, the C process, we posited in a very old paper in '93 that there was a nearest neighbor connectivity because the C process, this 1/f, we think essentially behaves like an isotropic process on the cortical surface. So it would reflect the eigenvalues that have been proposed more recently by Peter Robinson. With this basic structure of the 1/f activity and the alpha activity, we then constructed directed transfer functions. This allowed us to create a precision tensor, and that is, we have a precision matrix or inverse covariance matrix of the simulated EEG for each frequency. That's why it's a tensor, it's the precision tensor. And from there, we obtained an empirical covariance tensor. Now, this allowed us then to add to the simulated source activity. We could then add through the lead field, we would get sensor noise, we would get sensor time series with a power spectrum, which you can see here. And then we use this to compare with what is now practically a standard method, very much recommended for the analysis of magneto and electroencephalogram to eliminate leakage. And it is the method of working with orthogonalization of the time series to eliminate leakage and the use of the envelope of the complex Hilbert transform. So essentially the method, and Deirel can explain again and Riegel in more detail, we actually generated these, the complex Hilbert envelope covariance, the sensor narrowband covariance, and then we, as in the methods that are described, we applied both the standard, let's say, methods like E-Loretta and beamformer, as well as different varieties of our, let's say, sparse covariance graphical model estimator. And we use this for two models, one with a planar head, the other, which a planar head is simply what I call Donovan's brain. It's a brain that's floating in a tank of saline, so it has the simplest possible lead field. And then we have a realistic geometry obtained from one of our databases. And then here, the question is, how well do the different methods obtain or recover the precision matrix of sources? And I have to add that the standard method of orthogonalization and looking at the envelope of the covariance then has a further step of sparsification using the graphical lasso, whereas here it's done after all the calculations. We do it as part of the process of finding the sources and covariance, so it's integrated. The results, well, then this is the data analysis, simply, in which we compare both. I won't go into details. And here is the results for the planar or simple geometry for the lead field and the human. And here you see the simulated generators for the alpha rhythm, which is what we're focusing on. And you have where you see Higgs, that is the graphical model we have. And here you see that different varieties, and you see that we can get pretty good reconstruction. Actually, you can see down here more easily a numerical comparison in which we see the area under RC curve, sensitivity, specificity, precision, recall. In this radar diagram, the usual methods like E. Loretta, Roy, using the orthogonalization method, actually perform quite less well than any of the methods that are based on the graphical lasso method. And this is more accentuated when you have a realistic lead field. That is to say, when there's practically a very simplistic, infinite homogeneous lead field, they don't perform as badly, but the elimination of leakage is not as effective when you have a realistic lead field. We furthermore went on to test this in the model that we've and platform we've published for looking at the macaque brain with EcoG. This is work with Naotaka Fuji. This was already, we have data there. We have ECOG and simultaneous EEG, very sparse, only a few electrodes. What we found is that if we use a single layer ECOG lead field for the here, which we have to have because the ECOG also has a lead field. And here we have the EEG conductance model, which is more complete with several shells. And here you see where the electrodes are. Here we see the cross spectra at this peak here, at this peak here, for the sources using our source estimation method. And here, this is for the EEG. Now, the important thing here is, I'll summarize. You can look at this in the poster that the Riegel is presenting, and they can show you. Here, we have the same type of diagram where we're looking, but now at Kullback-Leibler distance, at the log-Euclidean distance, and the Riemannian distance and Riemannian geometry. So 3 different measures to compare covariances matrix to see the match between the source covariance that we found on the ECOG and the one reconstructed from the EEG cross spectrum. As you can see again, the usual 2-step methods actually don't perform very well, and as we're happy to say that our methods do perform quite better. Going further on, I have to tell you that this is part of an effort we're doing to do large-scale processing of large databases of EEG, which is part of the Cuba, China, Canada project, in which we are developing a series of toolboxes, and this is one of them, for use an analysis which will be completely open. It's not, all of the software is on GitHub, but we're preparing it also so that the data can be accessed through LORS. And we have, for example, prepared for early release all the Cuban human brain mapping database in the BIDS format to be released publicly also. We see here in the next slide the one of the toolboxes we incorporated, which is the methods we developed using the Cuban human brain mapping database, which has data from age 5 to 97 and can produce age-corrected maps of statistical parametric mapping of either voltages or sources. This is part of the effort. And then we see here the integrated pipeline. You can see here the databases that are already being prepared for public release. We have the Child Mind Institute, the Human Brain Healthy Brain Network. We have HCP, and we have this pipeline which we have prepared, of which the quantitative EEG I showed you and the new inverse methods I just also show you will be part. Now, we are preparing anatomical surface EEG and inverse solution. We're preparing fMRI, diffusion-weighted imaging, and anatomical. These pipelines will be SIFTI compatible. That's what we're producing now. And there's two objectives we want to achieve. One is to match this to the Big Brain project that's going on at the MNI. The second one is to provide links through narrow mass modeling to everything we're doing. This would provide a link to both SPM and the virtual brain. We have to say that we have now these pipelines that allow us in silico evaluation of fMRI, EEG, diffusion-weighted imaging. You see an example. We have now also addressing one of the main problems we're finding in processing thousands of EEGs, MRIs, which is quality control. We're developing robust semi-quantitative methods to detect problems. I can tell you, for example, that from the Child Mind Institute, 20% of lead fields end up by having serious problems. And what we're doing is to try to make this objective and to be able to do this in as automatic way as possible. The same for the recordings. Now, I mentioned the Big Brain. As you know, this was a joint effort by Alan Evans, Catherine Edmonds, and this Big Brain, which has been in the public domain for some time, has now been matched to the ICBM 152 template. So what we've done is to adapt our methods of inverse solution, including BC Vareta, and this is the first stage of BC Vareta, which I told you is the screening of active sources. And here you see that you have on the big brain template, and you have an individual subject from the HCP database. You have the different compartments, the placement of the sensors, as well as we're starting to compare matches between the reconstructed sources for the big brain template as well as for the individual template. And here we've done a collage where we have EEG sources for alpha. Here we have using the big brain template, and here we have magnetoencephalogram. Now, here you see in one particular case from the Human Connectome project and from the big brain template, the source reconstruction of the peak of the alpha. Now, this has to be bolstered and tied to neural mass theory. In this, we've just started this program with our young students, and you've seen there at the Congress, Ming Li and Anis. In the case of Ming Li, we've been working on looking for the effective dynamics of let's say of large-scale neural systems. And the idea is based that if you have a system of stochastic or random differential equations, in this case, we've worked as an example, the famous Jensen and Ritt model. We're working with a framework which was developed by the group by Koifman. And this group, in which we have Schnitzer, who has developed, is the idea that you can do it the same, you should go to look at the effective dynamics, the large-scale slow variables in the space which is spanned by the eigenfunctions, the continuous eigenfunctions of the backward Fokker-Planck equation of the system. Now, an essential point which we've already implemented and Ming Li has taken is down here that to do model identification, a Mahalanobis distance in data space approximates distance in the effective dynamical space, and then that means one can use the diffusion map methodology for manifold learning to find then the eigenvectors of these Mahalanobis distances to approximate the basis of the effective space. If you have this, you can then discretize the system and recover the system by continuous to discrete Kalman filtering or variational filtering. One can think here, we use the Kalman filter specifically because it's closer to the original work of the Koifman group. We think to modify it later on. Now, here are some of the results on the system verification. We have test and estimated signals. And what we did was to compute a dynamic time warping measurement of similarity, since let's say the speed along the manifold that's reconstructed not necessarily is the same as the one gets from the data. So here we see a quite good correspondence and separation. So a separation between this limit cycle behavior of the Jensen and Ritt model. You know that it has a Hopf bifurcation, and it also has a point attractor. So when one tests the similarity of the reconstruction between let's say the point attractor and the limit cycle, one sees that there's a clustering. It is very subtle. Both look like alpha rhythm, but as we demonstrated in 99, these two have quite different underlying dynamical behaviors. So this is one approach, but to be able to apply this approach and to be able to work with large-scale data, we need to be able to do this in an efficient manner. And in this, Anis Lady is working to develop an efficient algorithm for large-scale computation of neural masses. We know that the forward problem in any estimation is essential because it has to be repeated many times, no matter which method you use to do the inverse problem. So basically, it's the same model as before. Where Anis Lady is building upon is something that we described in 2009, which was to use random algebraic differential equation, in which we would use local linearization, and then this equality here, which can be solved for each neural mass separately in an explicit fashion. So we use Mathematica, and now Anis Lady has cleaned all this for public release using MooPad, so it's all in the MATLAB. We find the equations for the integration. We put this in then to the neural mass system, and this is all possible due to the existence of delays. Now, in my quarter lecture, many of you remembered that I said that connectivity was a tensor problem because you had the connection of sites versus time. And Anis Lady is building upon this tensor work so that we can now start putting in the delays not as a single delay, but as a continuous, let's say, distribution. As we know that probably delays are distributed according to certain factors related to axonal diameter, myelination, and so forth. And Anis Lady got to the first step of this, and you can see here a reconstruction that's already there with this delayed system.
Finally, I have to insert what we have been doing that goes beyond the Cuba-China-Canada collaboration, and in fact, a major consortium that we have created, of which Alan Evans and myself are the co-PIs, is the Global Brain Consortium, which groups many of the different large brain projects in the world and has a focus on using different imaging modalities to attempt to find biomarkers in the framework I outlined at the beginning of the lecture, but with an emphasis on electrophysiology as a technique that might provide an impact on global health indicators. The World Health Organization is part of this endeavor, and we recently had in Cuba a meeting, the second one, where we have outlined research programs to try to put all these ideas, actually to advance them and to extend them into actual trials in different economic settings. The knowledge that comes from the processing pipelines and methods that I've told may well be used to use brain-inspired or to develop brain-inspired technology. And I hope that this talk has given you an overview of this. And once again, thank you for inviting me to speak.
Good morning, everyone. My name is Athanan Wannachai. I come from Oasis Research Group, Chiang Mai University, Thailand. Today, I would like to present the real-time 7-segment display detection and recognition online system using CNN. The system is a machine status tracking system that can detect the machine display and interpret to the numerical form and show in the online display in real time. In the real world, the machine shows their status as the numerical data using the 7-segment display. We call in short is SSD. To track the working status of the machine, the human need to access the status data manually. However, in an action situation, the operators cannot monitor the machine all the time. The process information time lagging and human error may occur. The machine failure detection is delayed. The produced goods may have defected. The method to solve this problem, image recognition technique is applied. This technique can automatically obtain the working machine status without the machine modification. A digital camera is installed close to the manufacturing machine to capture the 7-segment display image. The images are analyzed and transformed into numerical data using an image processing algorithm. The problem of this research is the difficulty in the detection recognition process. The different kind of the machine, the camera module can install at the same position in all of machine. The view frame view difference and that make the capture image contain the varying angle and light disturbance. In this image, it shows the example display image with the skew image, blurring image, light disturbance image, and indicate background image. To finding the display screen automatically become a challenge in this case. In the research objective, to design a real-time tracking machine display online system, the system can be detected and recognizes the working status to the numerical data in the real time. We propose RSDC. RSDC is constant of the real-time 7-segment display detection and recognition online system using CNN. The ISDC is a consist of two parts, and the first part is the camera control module, and the second part is the interpretation of 7-segment display framework, as we call in short is ISS framework. This page is show the flow work of the ISDC. The camera control module capture the machine display and send the image file to the server via an internet, and the server get an image file and analyzes to the numerical data, the machine status as shown via the online display in real time. This page is show the first part. The first part is the camera control module. We use the one camera control module per one machine. The camera control module designed to capture the machine display light and send to the server every 30 seconds. This module is consist of the webcam camera and main control unit is the Raspberry Pi Generation 4 and power supply we can change the VAC to VDC to operate the Raspberry Pi and camera webcam. And the second part, it is the ISS framework. The server uses the ISS framework to detection and interpret the imported image. The output is the number or numerical data, and they are filtered before show the display online. IAS framework consists of three procedures. The first procedure is the SSD detection, and the second procedure is the SSD recognition, and the last procedure is post-processing. In the SSD detection and SSD recognition is applied the CNN technique that can detection and recognition the image file, and in the post-processing using adaptive bound criteria to reduce and cut the spike or noise in the output data from SSDR. We use CNN because the CNN is an artificial neural network, which is one of the bio-inspired algorithms. The CNN imitates the machine vision as a human vision. The human sees the image as a sub-area, and they combine the group of the sub-area to determine what they are seeing. This paper applies CNN technique to make the machine learn where the position of the machine display in an image, and interpret that image to the numerical data. CNN uses because the CNN technique that's suitable for the interpret an image to data, and CNN can automatically to find the different machine display without new configuration. Inside the CNN model consists of the two layers. The first layer is a convolution layer, and next layer is a fully connected layer. Inside any layer has many nodes. The node is trained by all datasets using the ReLU activation function. The dataset is the set of the classification numerical image from the detection procedure. The output of the convolution layer will be classified by the fully connected layer. That means the CNN that can detect the machine display where the machine display in the image, and the CNN model that can classify the value of the image number by learning the data set of the number. This is SSD detection workflow. The first step, get an input image and find the desired area with the CNN, and next, reduce the image noise. After clear the image noise in the process, SSD detection merging the input image with the desired area image. Now SSD detection can know where the machine display in an input image. The SSD detection crop only the image of the machine display and send to the SSD recognition procedure. The SSD recognition get an output image from SSD detection and find the digit by digit scanning process with the window sliding in the picture shown in the left window sliding. And SSD recognize using the CNN technique to define the number of each digit. The output of the SSD recognition procedure is the numerical data of the input image. The output data from SSDR that can plot in this graph. The blue line is the output of SSD recognition that we can call the raw data because they do not pass the post-processing in IAS framework. Sometimes the SSD detection and SSD recognition fail in the detection or recognition process that make a noise or spike. And to cut the noise, we cannot fix the bound size because we don't know the trend of the data or of the machine. The post-process apply the adaptive bound criteria method that can cut the noise and spike in the raw data any trend data set. The post-process is had a 4 step. First step is create the trend line. Second step is calculate the adaptive bound size and create the upper bound and lower bound is the third step. And the last step is to make the rule to cut the spikes. The first step, we create the trend line with the EWMA. It stands for the exponential weight moving average that you can follow in the first equation. It means CT. And we find the adaptive bound size by the second equation is in the picture show the beta T at time T. In the graph, you can saw the raw data is in the blue line and the trend line is the orange lines. The x-axis is the time in minute and the y-axis is the distance of the Y pooling. At time 23 and 39 time, SSDI is fell in the recognized an image that makes spike in this raw data in this graph. And after that, we know adaptive bound size. We plus and minus adaptive bound size with the trend line. As shown in the picture is in the yellow line and green line. Yellow line is mean upper bound. You can calculate by UT in the equation on the left. And lower bound is 2. Now we can saw in the graph about the raw data is the blue line, orange line is the trend line, upper bound and lower bound is a yellow and green. And the last step is we create the rule. If the raw data is in the bound, the data at time t, we using the raw data. But if the data out of the bound, the data at time t, we using the previous data to cut the noise or spike in this graph. In an experiment and results, we take the RSDC with the real image from the machine. The graph that shows the ground truth of the data of machine status. The ground truth created from the real machine display image, 200 image. The camera control module capture an image every 30 seconds. In the x-axis, we can get the time in the 100 time. The graph is the distance of the Y pooling. The Y pooling start at 0 meter and pooling in finish at the 400 meter. The worker reset the Y pooling and rework again every 400 meter. And this page is show the CNN parameter setup. The node in the convolution layer and fully connected layer is set up range between the 2 to 32 nodes per layer, but in this table it shows the base parameter of node, about 2 to 16 nodes per layer. The node is the length between the 2 to 60 in the first node and the second node, and the fully connected node is the base parameter is the 16 node, and node in the output layer is the 11 because the number of 0 to 9 and 1 is the space. This table shows the result of the ISS framework. This is the accuracy of the ISS framework with the base number set of any layer. The first column is the number of set of any layer in a number node in any layer. The layer 1 and the layer 2 is the convolution layer, and the layer C is mean the classification layer, fully connected. AC of ISS is mean the accuracy of the ISS before post-processing, and the column is the AC of the P dash P is mean the accuracy of ISS when passing the post-processing. The average accuracy of ISS is about 87.6%. 48%. But after post-processing, the average of the accuracy is up to 90.48%. And we calculate the standard deviation accuracy of ISS that equal the 6.5%. And after we post-processing, that can reduce the SD accuracy to 1.5%. In the conclusion, this paper proposed the real-time 7-segment and display detection and recognition using the CNN, as we call in the RSDC system. The RSDC can real-time tracking machine status. The system uses the camera control module to capture the 7-segment display and send the image to the server. And the server can use the ISS framework to real-time process an image into the numerical data and show it on the real-time display like that. The ISS framework is a high accuracy, about 80% when we use only CNN technique, and after we post-processing is up to 19%. That means the ISS is high accuracy to detection and recognition the machine display. Thank you very much. Have any question?
Hello everyone, this is Shanxue from Southeast University. Today my topic is a novel method for extracting high-quality RR intervals from noisy single-lead ECG signals. As we all know, ECG is one of the most important physiological signals. One of the important methods for analyzing ECG is about the heart rate variability, HRV. Then what's HRV? HRV refers to the dynamical changes of the difference of interbeat intervals. HRV analysis can reflect the stress, disease, and related pathological state of autonomic nervous system, and HRV features are changing along with disease and healthy aging, as well as different levels of physical activities. So, in order to get accurate HRV features, it is necessary to detect R peaks accurately in noisy ECG. In previous studies, plenty of high-accuracy R-peak detection methods were performed on ECG analysis. However, these excellent results were usually obtained from some standard and common databases. In these databases, the data quality of ECG recordings was pretty good, as shown in this picture. But when applying these detection methods on ECG collected in daily life or acquired from variable single-lead ECG devices, the R-peak detection results were usually unsatisfying. Due to the influence of data acquiring environment and devices, the collected ECG signals were often noisy, just like this picture. For noisy ECG analysis, the accuracy of R-peak detection method is difficult to reach the ideal values in the published papers. In order to achieve a higher accuracy of R-peak detection, we proposed a new R-peak detection method to synthesize the advantages of multiple detection methods. And aim of our study is to obtain more accurate R-peak detection results by combining 7 conventional R-peak detection methods from noisy ECG. So our methods consist of 5 steps. Step 1. The R-peaks of ECG samples were detected and labeled with 7 detection methods. Then we got 7 sets of initial detection results. Step 2. We categorize the ECG sample by template matching and select weights by the category. Step 3. We calculated the scores of each R-peak annotation in 7 sets of detection results in Step 1, based on the weights in Step 2. Then the scores were filtered by a threshold, and the annotations with raw scores were deleted. After this step, we got the preliminary R-peak detection results. And Step 4, according to the preliminary results of Step 3, the ECG sample was cut into multiple sections. And finally, we extracted waveform features from each section in Step 4. Based on these features, the quality scores of each section were calculated. The purpose of calculating the quality scores was to further eliminate wrong R-peak annotations. Here are the R-peak detection methods we used in our method. They are OKB, Pan-Tompkins, JQRS, NQRS, MTEO, phase-based QRS, and R detection by wavelet. These methods have been widely used in many studies, especially in single-lead ECG analysis. Due to time limit, the details of each method will not be introduced in my report. Please refer to our paper for more details. For details about the narrow R-peak detection method. First, the input ECG signal was analyzed by 7 R-peak detection methods mentioned in previous page to obtain 7 sets of detection results. Then, through template matching, ECG signals were classified into 7 categories, and each category was matched to a set of weights. Weight represents the ability of a detection method to detect R-peak correctly on a certain kind of ECG signal. Since I only have 15 minutes, the method for obtaining the weights is not detailed here, which was described in the paper. Then, the 7 sets of R-peak detection results were merged into a vector, and the scores of the initial R-peak annotations obtained in previous step were calculated one by one. So suppose we need to calculate the score of annotation J. The score was calculated based on the weight of the detection method that marked annotation J. Then find out whether there were other R-peak annotations marked by other 6 methods in the left or right range of annotation J. In our study, we set range to within 50 milliseconds around the center of annotation J. There would be some deviation between the results of several detection methods. So when many close annotations were marked within this range, it is likely that there was a correct R-peak annotation in this range. When other annotations within the range were more concentrated on annotation J, it is more likely that J was a correct R-peak annotation. So the score of annotation J was inversely related to the distance from it to other annotations within the range. And the score of RPIC annotation J was defined in expression 1. Finally, a threshold was used to filter out the RPIC annotations with high scores. And in previous steps, we synthesized 7 conventional RPIC detection methods and got the preliminary RPIC results, but in some cases, all the 7 methods made the same wrong detection, or some RPIC were all misdetected. So, to improve the detection accuracy, we extracted some features of ECG waveform for each heartbeat to calculate its quality score. The extracted features were curtices, skenesis, and frequency band power ratio. In the same way, we filter the detection results by the quality scores. After these steps, we got the final detection result. And we used 2 public databases to evaluate our novel RPIC detection method. The first database is the MIT BIH arrhythmia database, which is a commonly used database for new proposed RPIC detection algorithms. It contained 48 ECG records. Each of the 48 records is over 30 minutes and sampled at 360 Hz. Most of these records were obtained from inpatient, including different shapes of QRS complexes and many kinds of noises. The second database was downloaded from CPSC 2019, challenging QRS detection and heart rate estimation from ECG recordings. The training data consists of 2,000 single-lead ECG recordings collected from patients with some disease. Each of the recordings lasts for 10 seconds. The sampling rate is 500 Hz. This database contains noisy ECG and signals with different arrhythmia patterns. Because of the low signal-to-noise ratio, most of the commonly used RPIC detection algorithms were failed. So let's see how our method performed on these 2 databases. These are the representative results of the RPIC quality score of record 52 and 55 from CPSC 2019 training set. Red dots represent the R detection results of the proposed method. Black dots represent the reference label of RPIC annotations. The black number below the RPICs were the corresponding signal quality scores. You can see the parts we were marked are wrong annotations, and the scores of this part were lower than others. This is the performance of the proposed method on the MIT BIH arrhythmia database. It's obvious that the proposed method has the highest accuracy compared to the 7 detection methods separately. Among the results of other methods, the highest accuracy is 98.26%. And the accuracy of our method is 98.89%. It should be noted that the accuracies of some methods are not as high as that presented in the published papers, which may be due to different parameter settings or different data preprocessing. And this is the performance of the proposed method on the CPSC 2019. As we can see, in this noisy ECG database, all the 7 conventional methods performed worse than the proposed method. Other methods in MATLAB reported in the challenge. Our method achieved the accuracy of 55.47%, apparently exceeding the second place method's accuracy 40.73%, and very close to the best method's accuracy 57.32%. The rules of calculating accuracy used in CPSC 2019 were much more rigorous than the rules used in MIT BIH arrhythmia database, and the penalty for wrong detection was greater. So this was also one of the reasons for that the accuracy of this database are much lower than on MIT BIH arrhythmia database. In summary, we propose a narrow method combining 7 conventional outpeak detection methods to get high-quality RR intervals from noisy ECG signals. The detection results were obtained by assigning a weight to each method, and then the results were filtered by signal quality scores based on the ECG waveform features for further filter the annotations. From the evaluation results of 2 public databases, we proved that the proposed method effectively improved the accuracy of outpeak detection, and more accurate RR intervals can be extracted from noisy ECG signals. In addition to the 7 detection methods used in this study, we may add more available methods and do further comparisons to find the best combination in further study. That's all my report. Thank you.
Hi everybody, my name is Vinay Gautam. I am from Department of Computer Science, Aalto University, Finland. Today I am going to talk about leak-resistant design of DNA strand displacement systems. Contents of the talk are as follows. First I am going to introduce the DNA as a non-biological substrate for nanotechnology and 2 of its main branches, static versus dynamic DNA nanotechnology. Then I am going to talk about DNA strand displacement systems, leakages, leak-resistant DSD design, in which I am going to talk about 4 types of design building blocks: translator, OR gate, AND gate, bimolecular chemical reaction network, and then finally conclusion. So DNA is a biological material. It has peculiar properties, like it forms a double helical structure between 2 antiparallel strands. It follows a base pairing rule where A binds with T, G binds with C, and over 3 decades since the seminal work of Nadine Seaman, DNA has emerged as a suitable non-biological substrate for nanoengineering. DNA nanotechnology, the wider field, has 2 subfields. The first one is based on methodology self-assembly, in which DNA strands, for example here the sticky ends of 2 separate DNA molecules, are annealed to hybridize and form a stable binding. And this mechanism has been employed to design a variety of static nanostructures, such as one shown in this image is a cube assembled by Nadine Seaman in 2003 Nature paper. The second second branch in DNA nanotechnology, which is also the subject matter of today's talk, is DNA strand displacement mechanism, and the field itself is known as dynamic DNA nanotechnology. In this particular methodology, the DNA strands act as input as well as output, where a wider range of dynamic control is realized between a rationally designed system in which the underlying mechanism is a DNA two-hole mediated DNA strand displacement mechanism that we can see from here. In this example here, there are two DNA molecules. A single DNA strand has a two-hole binding domain XT star, and this has another longer domain which we call as branch migration domain, and then there is this partially double-stranded DNA molecule. The first step in this two-hole mediated DNA strand displacement is a two-hole binding, where short DNA sequences bind to the complementary DNA complementary domains, and the step is termed as two-hole binding, in which one the molecule has found a two-hole and there is a subsequent branch, complementary branch, in the partial duplex. Then this branch actually displaces the pre-existing DNA molecule, and so given an input in these two main steps, two-hole binding and branch migration, the output is released. So input as a signal and output as a signal. So this works a kind of a translator system in which one can design to implement cascades of DNA strand displacement systems. A variant of this mechanism is this, in which DNA, the partially duplex DNA molecule has an inactive two-hole on this side. So once this input molecule hybridizes by the two-hole binding, then the subsequent branch migration happens, and the molecule, the incumbent displacing molecule, is still bound by a two-hole domain. So then in the next spontaneous two-hole dissociation reaction, the output is dissociated. So there is a minor difference between this and the second reaction, is that the last step is a two-hole dissociation step. However, here it was a purely two-hole mediated reaction. So this step here is shown as a unidirectional reaction. However, this one is a bidirectional reaction, reversible reaction. So there has been a variety of implementations, in particular logic gates, and theoretically any CRN could be implemented. But here I'm going to talk about the logic gates implemented using this mechanism. So a AND logic gate, as we know that given two inputs, if both of the inputs are present, then the output is released. And in DNA strand displacement methodology, this could be implemented as with 3 shifts of DNA molecule, input is strand X, input is strand Y, and an AND gate complex which embeds the two-hole inactive toe-hole. So here the first molecule X comes in, opens up this green toe-hole, and then the second input comes in and then releases this output strand, which can further downstream participate in reporting process. The second gate, the second fundamental building block is OR logic gate, in which at least one of the inputs X and Y should be present to release the output strand, and this could be realized with this implementation where input X, input Y, and with these two DNA molecules that release independently. Given the input X, this can release output. Simultaneously, this can also release, so at least one of the two should be present to realize the OR gate. In theoretically, any logic circuits could be implemented by such a methodology, and here I present an example of such a logic circuit which was studied by Seeley et al. In 2006. It's a AND OR circuit in which a 6 input AND OR realization, which one can implement it as set of AND OR gates and corresponding input signals, and a final summing up AND gate which actually releases the final output strand that can be detected by a fluorescence mechanism. But coming to the practical side of such these implementation, the challenges these implementation face are about the leakages, the unwanted leakages that happen when these circuits are implemented in laboratory. And one such example with respect to the previous logic circuit is this fluorescence plot from the Seeling's 2006 paper, where if all the combinations of ABCD inputs are present, then the circuit produces the valid output. However, in cases where it shouldn't be active, it's still generating some output which are undesired. So due to spuriously generated displacement events, DSD logic gates sometimes release the output signals that we call as leakages. Eventual fraying, the opening of the blunt ends of these DNA, partially duplex DNA complexes, is one of the main constituent of these leakage mechanism. Let us see how these leakages are introduced. So here is this basic building block of these logic gates. There is no input here to displace this output signal here, but considering a scenario where the fraying has happened, so a couple of bases are opened up from this side, and if there is a complementary strand in some bigger complex available here, then it can eventually displace the output even in the absence of the input. And one can see that as this in the frayed of bases, the toe hole is bound, and then it can eventually displace the output. So it's a false output, which we term as leakage. So there has been efforts to design leak-resistant DST systems, and my paper actually proposes a methodology to design leak-resistant systems where the main building block that I propose is a 2-hole DNA complex. This complex here, so this complex actually has a robustness against the introduction of leakages, unwanted leakages. It has 2 open toe holes here, and it is formed out of 4 DNA strands, 1, 2, and a third which wraps around this, and a 4 here. It has a crossover domain, so that's a kind of a knot which has some binding between the strands, this wrapping around the strand and this one here. So it keeps the molecule in a particular tight bound location, and this is the input signal. So the methodology is based on the principle where this input has 2 toe holes here that are complementary to these toe holes here, and the method implements that once these 2 toe holes, so to remove this, to displace this wrapped around DNA strand, which is going to be participating in the downstream reactions, it requires actually binding it from both these toe hole domains, and the displacement event is successful only when the 2 toe holes are successfully bound and there is a subsequent branch migration in both of the arms. So let's see how it happens. So once this signal comes in here and both of it in a localized way, the toe holes bind, and then in subsequent branch migration in both arms, the signal after the branch migration is removed. So I use this as a design feature to actually implement resistance against the leakages in the DST system. Here one can see that the only binding points are available are toe hole 1 and toe hole 2. However, the other 2 which are not available for any kind of frame of. So let's look to the first implementation is about leak resistant translator design, where I implement a translator system. Translator is basically given a input X to produce output Y, and I use these TTDC complexes, 2 TTDC complexes, to implement this translator system. So input X and these 2 fuel complexes, the reaction proceeds as the first input X displaces this wrapped around signal, which produces an intermediate signal this and a waste product here. The intermediate strand again participates with reaction with second fuel complex and displaces the output signal finally. So this realizes the translator system. Similarly, I also propose the implementation of this OR logic. So in OR logic, as we know that at least one of the X and Y should be present to displace to release the output, and this could be realized with a set of fuel complexes and the 2 inputs, and the block diagram of this can be seen from here. So they are basically the first 2 translator. Basically this block implements the OR gates. So up to here the OR gate is implemented, realized, but as we needed independence of the output signal from the input signal, so then another translator stage is added here. Likewise, an AND gate could be realized with this in this methodology, where I implement it as with the help of another design of the translator that I term as integrator translator, in which once the input X displaces this signal, then the toe hold is open, which again then the molecule actually reacts, the input Y reacts with this and displaces the output. So it implements the output, the AND logic here in 2 steps. Yes. And then this generic reaction of leak resistant bimolecular chemical reaction implementation, I also present this as a generic reaction, so that in principle any reaction can be implemented using such a methodology. So I implemented with the help of 6 fuel complexes, 1 integrator and 5 translator molecules with X and Y. So then the plug diagram is shown here. Finally, coming to the conclusion, so this methodology is actually quite generic, and one can use it to implement any kind of logic circuits, and also in principle any chemical reaction network can be converted to the equivalent DNA strand displacement implementation, and methodology can be implemented for the leak resistant design. And I am further looking at the future work to quantitatively analyze these errors. These are the references. Thank you very much.
Hello, everybody. I am Ward Fadel, a PhD student at the Faculty of Information Technology and Bionics at Pázmány Péter Catholic University in Budapest, and at the Institute of Cognitive Neuroscience and Psychology, the Research Center for Natural Sciences at Ulbert Lab, where we are conducting brain-computer interface research. I will present our work, which is titled Chessboard EEG Images Classification for Brain-Computer Interface Systems using Deep Neural Network. And this work was done by me and my colleagues, Moots, Csaba, Gege, and Professor Ulbert. First, I will start with an introduction about brain-computer interface systems, then a short review about transforming motor imagery EEG signals into images, then the materials and methods that were used. After that, so the data set, which is the Physiobank dataset, a widely used dataset in this research field. Then we propose our method, the chessboard transformation, to transform the motor imagery EEG signals into images in order to be classified using a deep recurrent convolutional neural network. And then the azimuthal projection with Klaftacher interpolation was also used to transform the EEG signals into images, just as a comparison with the chessboard, to compare it with the chessboard images. Chessboard transformation. After that, the results, and finally conclusion and our future plans. So the online brain-computer interface system is generally a closed loop, starting with EEG signals produced by the subject and gathered from electrodes over the scalp. More specifically, we are interested in motor imaginary-based brain-computer interface systems, where the subject is asked to imagine moving his hand or his right hand, left hand, the tongue or feet. So he's asked to imagine moving a body organ, and this imaginary movement modulates brain activity, especially in mu and beta bands. So usually this method is used with people who suffer from motor disability. After that, these gathered EEG signals are pre-processed, and then the features are extracted. After that, appropriate features are selected, then classified in order to be translated into commands for an application. Finally, feedback is provided to the subjects containing information whether their mental commands were recognized or not. The classification of the motor imagery EEG features is the main part of brain-computer interface system, but it's a challenging task because the EEG signals are noisy, so the signal-to-noise ratio is low, and they are non-stationary signals. They differ from one subject to another, and even for the same subject from one session to another, and they are highly sensitive to artifacts. So this is on one hand. On the other hand, it's a time-consuming task to record EEG data, to prepare the subjects and the material and the devices and the already designed room for recordings, and generally to train a model it needs large data sets. So this makes it a challenging task to classify the EEG signals. So the neural networks approach, the deep learning, has been investigated thoroughly and outperformed many other approaches in computer vision, natural language processing, and robotics, but it is still not investigated enough for motor imagery EEG signals classification, because the network performance strongly depends on the network, on the model, on the architecture, on the metrics, and large input data is needed, which is, as mentioned earlier, it's not the case for EEG signals. The EEG input to this network, to the neural network, can be either the raw data, the raw EEG signals, or extracted features, for example, using principal component analysis, or images. And actually transforming the EEG signals into images is getting more and more research interest. Usually researchers transform these motor imagery EEG signals into images by using traditional techniques like fast Fourier transform, short time Fourier transform, continuous wavelet transform, or scalograms, and by using only a small set of electrodes, like using only C3, C4, and CZ electrodes, and using somehow small data sets. So in order to overcome some of the shortcomings of these researchers, we use the PhysioNet motor imagery EEG dataset, which is based on the 1010 system with 64 electrodes. So first we use the whole set of 64 electrodes to obtain the data, because as we mentioned earlier, as those signals are so sensitive and noisy and differ from one subject to another and for the same subject, so sometimes if we choose only a small set of electrodes, it's not working for a specific subject. So for instance, maybe this subject needs additional electrodes in order to obtain the best features to be classified. And relatively, it's a large data set with 109 subjects. We actually used 103 subjects because 6 subjects had bad recordings, and each subject is performing 14 experimental runs, 2 baseline runs, 6 real movement runs, and 6 imagined movement runs. And we are interested in the imagined movements because the goal is to classify the motor imagery tasks. So each run has 30 tasks, and each task lasts for 4 seconds. And we extracted those motor imagery tasks, and they were labeled accordingly. So those tasks are like, the subject is asked to imagine opening and closing his right fist, or imagine opening and closing his left fist, or both fists, or both feet. And after each active task, like after each imagination task, there is a rest or relax. The subject is asked to relax for 4 seconds, then to imagine something for 4 seconds, then to relax for 4 seconds, and etc. So the goal is to classify these tasks, those 5 classes. We propose the chessboard transformation, and it works as the following. So first, the labeled rest and motor imagery tasks, which were obtained from the previous stage, they are filtered into mu and beta bands, because as we mentioned earlier, the motor imagery activity hides here. And then 0.4 seconds window fast Fourier transform is applied over each 4 seconds trial, so we get 10 different measures over each trial. Then the sum of the squared absolute values was calculated for each electrode, and this actually will be represented as color intensity. So then the chessboard projection comes. The electrodes in the 3D space are projected into a 2D plane, and each electrode position is represented as 4 by 4 pixels square. The color intensity of each 4 by 4 pixels square represents the activity power obtained from the corresponding electrode, where green grades represents mu band activity and red grades represent beta band activity, and thus we got 32 by 32 unicolor images pairs that represent the 64 electrodes activity of either mu or beta bands, and by adding them together we get the 2-channel images. So here are the 2-channel images, which is 32 by 32 images, and each one is represented 4 by 4 pixels, and it represents the an average activity over 0.4 seconds. So we get 10 of these images for each trial. So the images per subject, we have 30 tasks, and for each task we get 10 images, and each subject is performing 6 runs, so we have 1,800 images, and for 103 subjects we have 185,400 images, which is relatively good to train a deep neural network. Now the neural network model. So we used a CNN, convolutional neural network, followed by long short-term memory. The CNN is used to extract the spatial and frequency features of the consecutive EEG chessboard images, while the LSTM is used to extract time dependency, the long short-term memory, to extract time dependency between consecutive images. So between consecutive EEG images. So this model is generally used for video classification, and now we used it to classify the EEG chessboard images. So the problem of classifying EEG signals becomes like a video classification or image classification problem, where neural networks or where deep learning is good at. The CNN structure is as the following. We tried different architectures and with different parameters, and the LSTM is a 1-layer with 128 cells. After that, fully connected layer and softmax layer, in order to classify the 5 already mentioned classes. On the other hand, Here is the azimuthal projection with the Clough-Toucher interpolation. We used this approach in our previous work, and now we are comparing it with the chessboard approach. So the first steps were as previous. So we obtained the 5 classes, then fast Fourier transform, then sum of absolute values, and then it comes the azimuthal equidistant projection. So first it's equidistant projection, like the position of each projected electrode, each of the projected electrodes to a plane that is tangent to the head top point are equidistant relative to that point compared with the original 3D space. So in a way or another, it's keeping the distances between the electrodes, and then Clough-Toucher interpolation was used to generate the two-channel images. So the spatial and the frequency features are preserved, and the input images are the same as because the same paradigm was used. So they are the same, has the same number as the one with the chessboard approach. Now to compare the results, we use the leave one out cross validation, where one subject is used, I mean the whole 1,800 images for one subject, randomly chosen subject, are used for testing, and again another subject data are used for validation, and the rest 101 subjects are used as a training set. So each time we are choosing randomly one to test and randomly one for validation, and the rest 101 subjects for training, and we obtain the results for each subject, and also the average results, I mean for the 103 subjects. So the results for the chessboard approach were better than the azimuthal projection approach when the two-band scenario was applied, and also they are compared with the support vector machine, which is a baseline method when it comes to motor imagery EEG signals classification. And actually also those results could be compared with the state-of-the-art methods. It's important to mention that now here are the the subjects with the highest classification accuracy, here for the chessboard approach and here for the azimuthal projection approach. So we can notice the following, that first, for some subjects it was high for both approaches, but for some subjects, like subject number 21, the classification accuracy was high for the chessboard transformation approach, but it was relatively low when it comes to the azimuthal projection approach. And this actually leads us, and actually for other subjects it was vice versa. So it was low for the chessboard approach and high for the azimuthal projection approach. So this led us to the fact that transforming the method of transforming the EEG signals into images play an important role in classification results. So for this subject, it's hard to tell by applying only one technique or one network configuration to tell whether his classification accuracy is high or not. So it's better to apply different classification techniques and then to tell the trend about each subject. So this is here when it comes. Also, it's very important to mention that when changing the network structure, also these results were changing, and when changing also the parameters, and when so the results are really highly dependent, first of all, on the technique that is used to transform the EEG signals into images, and secondly on the network model and the structure. So now here we proposed, now the conclusion and the future work, we proposed the chessboard motor imagery EEG signal to image transformation, which was effective and superior to other methods. A deep convolutional neural network model was applied, as mentioned previously, and I guess it's important to say that transforming EEG signals into images is a good direction, is a good research direction. The results are promising and could be compared to the state-of-the-art results, and the classification, as we mentioned, it's highly dependent on so many factors. As a future work, we are planning to test the model on different data sets, especially the one we are recording now with high-tech equipment at Ulbert Lab, MTATTK, and you can visit us and see what we are doing there. And also we will test different signal-to-image transformation methods. So we are already now applying another good approach, but it's not mentioned here. So it's very important to mention that changing the methodology might bring great results. So it's a hot research area. Of course, we will test different network models like GoogleNet or whatever, and finally applying a channel selection techniques. So to reduce the number of electrodes, not to use the full set, the 64 electrodes, which might also bring better results. Finally, thank you for your attention. This is my email for any questions or for any research collaboration, and this is our lab. You are invited to check and to contact, and thank you so much.
My name is Liao Yinhua. I'm from School of Information and Communication Engineer, University of Electric Science and Technology of China. I'm very pleased to join the France. The topic of my presentation is target tracking based on DDPG in wireless sensor network. My presentation will include these 5 parts. First, some background information about the research. Second, the main work we have done. Third, the specific research process. First, data organization and calculation. The lesson innovation and presentation of my published paper. First, I will briefly introduce the research background. Wireless sensor network have been deployment for more than 30 years. It consists of a large number of tiny nodes with sensing, computation, and wireless communication capability. Its purpose is to monitor the environment rather than communication. So, target detection and tracking are classical applications of motion critical sensor and sensor network. With the rapid development of technology, motion critical sensor and sensor network have some unique features compared with ordinary wireless sensor network. Their following challenges are recorded as a key instrument to work out, such as the X in relation, bandwidth utilization in mission critical oriented wireless sensor network, and high length response and feedback, and so on. However, most concrete research about mobile mission critical sensor and sensor network focus on increase the coverage, identify routine hotel or operation data collection, and for improve the accurate detection and real-time tracking are very limited. Therefore, combining reinforced learning to operate the node selection range of ticket tracking. Deep deterministic police gradients is reinforced learning framework that can handle the continuous action space based on the actor critical growth. We can see the picture. It includes actor network and critic network. The DDPG have the critic and actor network. Its parser and update process are inactive. We will describe the following format. It's the police update granted. The actor net update the police with the aid of the credit network. Next, I will introduce the main work of this paper. The main work we have done can be summarized as 4 parts. First is the deployment about the loads in the range. Second is the trajectory prediction. The third is we use the DDPG to solve the convergence. We know when we track the target, it's no convergence object. So we use DDPG to solve this dilemma. Finally, we show the simulation to glass. Then I will briefly introduce this research. First, I define the deployment mobile and the data mobile. I define the deployment model and the data model. In the ranging, we deploy 2 kinds of load, static load and mobile load. The static load on the edge of the meter edge. The mobile load is inter. And the interment dispatch central mobile load to track the target. Second, we divide the mobile loads into 4 working states, include idle, sleep, check, work, and work. The 4 states have different energy consumption and detection frequency is so we show the performance in terms among them. About include the tracked occurrence, energy conference, and the rate. About them, the tracking response time is not least, and is set to a value inverse propagation to the load screen center to train the mobile load to track the target. And this reward is the key value of the load to train the mobile load to in the track the target. It is relative to the energy components of the track process and the tracking response time. Then I will introduce the tracking process. It may include have two steps: placing and tracking. We use the CARM filter to break the trajectory and then expand banish mobile to run as the target of the target by motor load. We can see the exam flow. It's use DDPG to select the operative mobile load to caster. The algorithm flow, which is not much different from the DDPG algorithm, mainly because the reward maximum and string update are inbound. Then we use the Parson and the TensorFlow deployment to context simulation experience. Select 100 internal mobile loads and 40 for endage static loads, and a target among them. The simulation patterns are set as follows, and the summary interpretation as follows. Follows the comparative activities, the experiment is mainly compared with the random selection system to verse the relationship of the algorithm. Many increase take transportation errors, take loss rates, cumulative error, average system cost. We can draw from the following calculation from this picture. The first picture is target node tracking error for one iteration. It can be seen as the time increase, the trajectory prediction error of the target nodes becomes smaller and smaller, which can ensure the contribution of each node is more readable internally. The second is target loss rate of the irregular pattern with by different secure joining schemes. We can see the random selection scheduling schemes don't perform well, since the strain selection of sensor is low, because there is no learning choice. The track train is by the different sensor. We can see the take north rate of the interagement security schemes currently decrease with the increased weight of the number of interactions and reach a century level of 1 0 point 0.2. There may be room for optimization in the future. The third picture is cumulative arrange tracking error by different sequential tuning segments. It shows the comparison of the cumulative system tracking error of different sequencing schemes based on DDPG learning scoring. We can see with the iteration, our purpose schemes can increase tracking occurrence 33.5 points approximately compared with the random selection security schemes. The last picture is a range system cost achieved by different tracking occurrence. We compare the cumulative system cost achieved by different tracking occurrences in intelligent sequence schemes. It's less than 5 meters, 8 meters, and 10 meters. It can be seen that when the target tracking stream is high, the warning is small. In general, this paper, we can get the involvement. First, a lower comparative mobile system based on DDPG for wireless system for motion critic sensor and sensor network. Track tickets consists of strand selection layer and differencing strand. Second, we post a stem normalization algorithm to make the train much faster and easier. The last, motion critic sensor and sensor network based on DDPG algorithm can learn continuous technical tracking process through training and audio mode without a large number of technical file post-dates. That's all. Thanks.
Hello everyone. It is my great honor to give you a report on this meeting. I am a graduate student of University of Electronic Science and Technology of China.
My research field is wireless sensor network. My paper's title is A Fuzzy Tree System Based on Cuckoo Search Algorithm for Target Tracking in Wireless Sensor Network. I will elaborate from the following aspects. First, let me introduce the wireless sensor network. WSN is a kind of distributed sensor network. Its terminal is the sensors that can sense and track the actual world. WSA sensors communicate through wireless mode, so the network settings can be flexible and device location can be changed at any time, and can also connect with the Internet in wired or wireless mode. Generally speaking, a wireless sensor network consists of small nodes that have communication, computation, and sensing capabilities, which is now among the most essential technologies. The sensor nodes are limited in terms of memory, energy, range of transmission, and processing capability, with the ability of connecting diverse devices with each other. Internet of Things (IoT) has been considered as a key technology for the next generation mobile networks. In the past few years, the application of WSN has been more and more extensive, such as navigation, tracking, virtual reality, and other technologies. Let me see in the picture. There are applications of WSN which all need the location information of the nodes. So the position of mobile nodes has become an important research of WSN. Next, let me introduce the target tracking in WSN. Mobile wireless sensor network is a special kind of IoT. In WSN, sensor nodes can monitor surrounding areas to guarantee environmental safety, and they can achieve critical tasks through monitoring moving targets. Monitoring data is collected and forwarded to the sync node or remote cloud. The sensor nodes carried about the corresponding actions according to the feedback from the cloud or the sync node. In this picture, the sync is here and the target is there. Next, I will introduce the cuckoo search algorithm called CS algorithm, which was proposed in 2009. The cuckoo search algorithm is based on the breeding characteristics of cuckoo. Cuckoo, which in the picture is a kind of magical bird, not only because of its beautiful singing, but also because of its active breeding strategy. Cuckoo put their eggs in the nests of other birds. Researchers describe the CS algorithm by 3 rules. Let us see the rules. Okay, the first one is each cuckoo lays 1 egg at a time and dumps its eggs in a randomly chosen nest. Second, the best nests with high-quality eggs will be carried over to the next generations. Third, the number of available host nests is fixed, and the bird laid by a cuckoo is discovered by the first bird with a probability. In this case, the host bird can either get rid of the egg, or simply abandon the nest and build a new nest. Okay, next I will introduce the genetic algorithm. Genetic algorithm, which called GE algorithm, is a method to search the optimal solution by simulating the natural evolution process. Genetic algorithm starts from a population which represents the potential solution set of the problem, and a population is composed of a certain number of individuals encoded by gene. This is the composition of genetic algorithm. And the next one is fuzzy logic. Fuzzy logic. Some problems with fuzzy concepts such as far, near, last, slow, and so on are similar, which means that there is no absolute standard for people so-called far and near. That is, we don't know how much greater is far or whether it is near. However, machine thinking is based on such discrete rules, that is, not far is near. Let us see the example. Okay. There is no strict boundary between heavy rain, light rain, and moderate rain. That is to say, the size of a certain rainfall does not belong to a certain category, but it is measured by membership. As we can see in this picture, the membership is shown in the picture. For example, for 10 mm rainfall, the membership degree of light rain is 0.5, medium rain is 0.4, and the heavy rain is 0.1. For 100 mm rainfall, the membership degree of light rain is 0, and the medium rain is 0.3, and the heavy rain is 0.7. The next where I will introduce the system model which I made. In this section, all the simulation experiments are carried out by using MATLAB. The total number of sensor nodes, namely node density, is set to be 30, 40, 50, and 60. Here, we chose the number of mobile nodes based on the density of the distribution of mobile nodes. As we all know, the more the number of mobile nodes is, the better the tracking accuracy will be, but it will increase the network energy consumption, so it is very necessary for us to select the appropriate number of the mobile nodes. We compared CS and GA algorithm from the multiple perspectives. The target moves in line with V equals to be 5 mile per second. Okay, let us see the solutions. From the two pictures, we can see that the CA is much better than GA in reducing the target error rate, and when n equals to 60, m, which means moving nodes for target tracking, equals to 2, the tracking effect is very good, and the calculation cost is less. Next, we have a comparison between CS and GE on the loss rate of the target. Okay, let us see the third and the fourth picture. Compared with the GA algorithm, CS algorithm indeed reduced the target loss rate, but the effect is not very obvious. When the deployed sensor is increased to 60, the effect of the two algorithms is similar, as we can see in the picture. But in terms of running time, CS takes less computing time than GA, which we can see in the picture 5. As we all know, time consuming is a very important measurement for a network. From this point of view, CS has more advantages than GA. This is the picture 5. From the picture, we can see that CS takes less computing time than GA algorithm, which can be used widely. Okay, let me do the conclusion. Compared with CS and GA algorithm, the CS algorithm performs better in terms of reducing average location error and increasing the convergence rate, which can reduce the consumption and prolong the lifetime of sensor nodes. The effectiveness of the CS algorithm has varied through simulation results. And that is my presentation. I'm sure that the CS can be used widely in the AI, IoT, and 5G aspects. Okay, thank you very much.
Hello everyone, today we will talk about this paper named Sensor Scheme for Target Tracking in Mobile Sensor Networks. We will talk about 6 parts. The first part, the problem this paper researched. Target tracking is an important application of wireless sensor networks. Compared with traditional WSN, mobile sensor networks has the advantages of strong robustness, flexibility, energy saving, and so on. In recent years, people have begun to study MSN. This paper investigates the extended Kalman filter algorithm in MSN and proposed a low energy consumption, high accuracy sensor scheduling strategy to perform the target tracking task based on EKF algorithm. The second part is the significance of this research. With the rapid development of wireless communication technology, WSN has been widely used in the fields of national defense, environmental protection, and building monitoring. In the era of 5G, WSN will play a crucial role in the Internet of Things. Many disadvantages of WSN gradually exposed. For example, due to the inflexibility of the network, some sensors will continue to play the role of network hubs, accelerating the exhaustion of their life. In addition, since WSN is a full coverage mode, the energy consumption is greatly increased. MSN is often combined by static sensors and mobile sensors. The introduction of mobile sensors has greatly extended the average life of sensors and enhanced the robustness of the network. Target tracking is an important application of WSN. For MSN, the introduction of mobile sensors brings more considerations, such as heterogeneous network processing, sensor path planning, sensor selection, and so on. In this case, we need to design the corresponding target tracking algorithm or strategy so as to achieve the requirements of energy saving and high accuracy. And then we will talk about two previous works in detail. The first one proposed two multi-load joint movement algorithms based on neural network and fuzzy genetic tree. The two algorithms find the current time optimal sensor selection and path planning scheme by training the historical samples. Its shortcoming is that they adopted two kinds of algorithms, combining machine learning and heuristic algorithm, which will greatly increase the complexity of the system, may result in unable to real-time tracking, especially often the system needs to consider concept drift and the model could be updated in real time. It can hardly achieve for distributed WSN, which is also very demanding for centralized network of computing power. The second one is proposed an improvement EKF algorithm. They give a new real meaning to the Kalman gain matrix, and they modify the original objective function of the EKF algorithm. Introduce a penalty term as below, and then they solve the new objective function by using the ADMM algorithm to obtain the Kalman gain matrix. And its shortcoming is that they directly use the load scheduling into the original EKF algorithm process and change the original algorithm structure. In fact, such load selection and path planning are not reliable. The EKF algorithm is a relatively sensitive algorithm. When the nonlinearity of the system increases, the EKF algorithm is easy to diverge. This approach will cause the EKF algorithm not to obtain the minimum value of trajectory prediction, but to obtain the minimum value of the objective function construct in this paper, which may also cause the EKF algorithm to diverge. Therefore, we need to perform mobile sensor selection and path planning independently from the EKF algorithm. And then we will talk about the idea of this paper. Considering the increased computational complexity of machine learning algorithm combined with heuristic optimization algorithm, this paper chose a more lightweight EKF algorithm for target prediction. However, unlike the idea of the second reference, our approach is to separate separate the EKF algorithm from the mobile sensor selection and path planning process without interfering with each other. In this case, the Kalman gain matrix is still optimal, that is, the target prediction error is minimal. At the same time, the selection of mobile sensors and path planning are determined by independent strategies. And our system can divide it into 5 stages. First is system modeling, and then EKF algorithm prediction, and then mobile load selection and path planning, and then EKF algorithm update, and then we will do system simulation. First, we will introduce the EKF algorithm framework. The EKF algorithm this article used is a nonlinear measurement model, but a linear target movement model. This model can be written as this one, and F is the transition matrix, and Hx is a non-linear measurement function. Omega k is the system noise and satisfies the Gaussian distribution of zero mean, and covariance matrix is Qk. And Vk is the measurement noise and obeys the Gaussian distribution, which satisfies the zero means, and covariance matrix is Rk. So we can write the EKF algorithm framework. The first stage is prediction, and the second stage is update. And then we will do the model deployment. In this paper, the monitoring area is a rectangular area of size b times b square meters, and assuming a total of n mobile sensors in the air, their initial deployment position are uniform, as shown in Figure 1. In fact, this initial deployment assumption is arbitrary, and it has no effect on the target tracking algorithm proposed in this paper. Also, this article considers heterogeneous networks, that is, sensors in this area are of different types. Suppose there are n sensors, and their 4 main parameters are considered in this network. First is the remaining power, and the second is sensing range, and the third one is the moving rate, and the fourth one is the current position of the sensor. We assume that a moving target appears in the monitoring area with a certain trajectory, and the first value of the trajectory is used as the lower initial state value. The target will be detected its position information xt, yt by some sensors at time t during the movement. These sensors will send their measurements to data center, and then data center use EKF algorithm to estimate the target trajectory, which means to predict the position and the speed of the target at the next moment. At the same time, data center will select 3 optimal sensors according to the current sensor distribution, plan the movement path of each sensor, and then perform target tracking. This is the initial sensor distribution, and the dotted line circle represents the sensor's sense range. And at time t, the motion state of target can be represented by this one xt, and we construct f and q. And at time t, it is assumed that the measurement vector after fusing of all sensor data is zt, and for each component zti, which is the measurement value corresponding to the ith sensor, can be expressed as this one, and the P0 represents the signal power transmitted by the target, and the DTI is the distance between the eye sensor and the target. And the DTI can be calculated as this one. After the basic model in the EKF system has been completed, the target trajectory prediction can be achieved. Then we need to design the sensor selection and moving strategies. As mentioned before, we need to perform mobile load selection and path planning independently from the EKF algorithm. First is load selection for sensor load Si. Its state can be represented by this table. We assume that the load's energy consumption is proportional to the displacement of the load. We score all sensors based on strategy 1 at each design moment and then select 3 sensors with highest scores for target tracking. If the current energy of our sensor is no longer sufficient for the next mobile tracking task, we consider it to be exhausted and set the score to the lowest. And this is strategy 1 for used for sensor selection. And we will talk about its idea. The absolute maximum detectable distance Rmax of our sensor is defined as this one. And the idea of scoring is to judge whether the sensor can capture the target at the next moment by the difference between Rmax of the sensor and the target distance at the current moment. And the sensor that can capture the target is superior. In order to evaluate the sensors with different Rmax, we need to normalize them. The normalized minimum score is -1. And after the sensors are selected according to strategy 1, strategy 2 will be introduced next. Strategy 2 makes the selected load move properly so that the target location can be complete and the location is used as a feedback to EKF algorithm. And this is the strategy 2 for used for sensor moving. And then we will talk about its idea. The idea of the moving strategy is that if the target appears within the half of R range of the sensor can remain stationary to achieve energy saving. If the target is within half of R to 2/3 of R, the sensor's moving direction is the moving direction of the target's current time and previous time. By doing that, the detection coverage area of the selected sensor can be kept largely. If the target exceeds the 2/3 of R range, and the sensor load will be in danger of not being able to detect the target at the next time. So they must move towards the target at the next moment. And this is the whole processing for this system. And the first is initial parameters, and then predictor process, and then sensor selection, and then sensors moving, and the last one is after the process. And then we will talk about the system simulation. And we deployment MSN to monitor air, and any people entering this air is considered as a target. And each sensor is regarded as a mini car. The task of the MSN is to track the intruding people in real time. Then we initial the parameters, and we consider the RMSE as the tracking error. Totally, we use 3 kinds of target trajectory to simulate: the power trajectory, the square trajectory, the circle trajectory. For each trajectory, EKF algorithm in static WSN is compared with this system. The first one is power trajectory. The left picture is the trajectory prediction, and the right picture is the two-thirds of track the two-thirds of track of RMSE, and the yellow the green line is the static WSN, and the red one is the dynamic MSN. And this is the square trajectory, and this is the circle trajectory. As we can see, the red one is always lower than the green one. So from the three kinds of target trajectories simulation results, we can see that although static EKF system is fully covered, dynamic EKF system performs much better. These results reveal that our strategies make the EKF system obtain a higher accuracy, while it only schedules 3 sensors to move to track the target, which is more energy saving compared to static WSN. And then we will talk about the future work. EKF algorithm can be applied to weak nonlinear systems, but in strong nonlinear systems, a huge flaw is that the first order linearization approximation of the system will bring large errors in state estimation, and EKF algorithm may diverge at this time. In view of this problem, the future research direction may be more inclined to adopt the algorithms which approximates the posterior probability density distribution of nonlinear functions such as UKF and PF. At the same time, the method of combining machine learning with heuristic optimization algorithm can be considered, such as frequent pattern mining and sequential pattern mining, which can be used to predict the target trajectory. Okay, that's all. Thank you.
Hello, this is Chang Lee from Yonsei University. I am going to introduce our novel Testbed, molecular MIMO communication platform with BTSK for embedded network systems. We target to build communication systems where RF communication is hard to utilize, such as the human body network. So, we have developed the testbed to mimic a blood vessel network. But molecular communication has low transmission speed compared with traditional communication. So, we applied proposed MIMO technique to increase transmission speed. This is the whole figure of the testbed. As I talked before, we tried to mimic the blood vessel network. So the pumps work as the heart and the tube network acts as the blood vessel network. The whole process of the platform is like this. The pumps make a flow and send molecules according to the transmitting algorithm. The sending molecules make a change of pH value at the receiver. The pH sensors measure the change and convey the signal to the computer. The computer detects the signal and visualizes the result. I am going to introduce each part of the testbed in detail. This is the concept figure of the platform. First, transmitter. The transmitter consists of 4 water circulator pumps and a microcontroller to control the transmitter and reservoir for chemicals, acid and bases. Next is channel. The channel consists of several lengths of tubes and 2 types of junction parts, which is made by a 3D printer. Therefore, we can configure various and complex tube networks to mimic a blood-based network. Receiver. The receiver consists of 2 pH sensors for measuring pH value, microcontroller to convey electric signals from pH sensor to a computer, and a computer for detecting the receiving signals and visualizing the receiving sequence. System operation. The transmitting sequence is divided into 2 parts and encoded to be sequenced by International Telegraph Alphabet Number 2, ITA2. ITA2 encodes alphabet to 5 bits. For example, alphabet B is encoded by 10011. The testbed transmits the coded speed through transmit antenna, which is pump 1 and 2 simultaneously. It utilizes the spatial domain for enhancing the transmission speed. We use binary timing shift scheme BTSK modulation to convert bit sequence to the chemical signals. BTSK modulation divides a symbol into 2 periods and modulates information by the index of molecular emitting period. The transmitter sends molecules at the first slot of the symbol when input is a bit 1. On the other hand, it sends molecules at the second slot when input is a bit 0. The platform can utilize both acid and bases as an information carrier. However, this time we use pH 4 standard solution. At the receiver, Two pitch sensors measure pitch value change by time, and the computer detects the transit signal at each symbol duration. The receiver utilizes an adaptive thresholding algorithm, which detects 0 or 1 by the threshold value. The threshold value is determined by experimental results and channel state information. I am going to show you a video that summarizes our platform. Our platform is a preliminary version, so it has some limitations. The main issue is increased error. The slow change of pitch value at the receiver influences to the error. We infer two factors cause the slow change. First is the shape of the receiver, and the second is bad performance of pitch sensor. The sensor is under $30, so we expect that we can enhance the performance if we use a more expensive sensor. To solve the error, we are going to making an optimal shape of the receiver. And we are also considering to compensate for the pitch sensor performance by adapting its measure process. Thank you for listening, and please leave a question if you have.
Hello everyone, this is Görkem Durmaz from Boğaziçi University Computer Engineering Department in Istanbul. Thank you for participating. I'm going to talk about our work, preliminary studies on flow-assisted propagation of fluorescent microbeads in microfluidic channels for molecular communication systems. Here is the outline. First, we start with a brief introduction. In molecular communications, chemical signals, organic and inorganic molecules, are utilized to transmit and receive information between devices. These devices are scaled in nanometric range and called nanomachines. Nanomachines can communicate with each other for cooperation to perform complex tasks. A network of nanomachines can be used in different applications, such as catalysis, sensing, electronics, magnetics, photonics, and biomedicine. However, building such a network is not an easy task, since there are multiple challenges in physical layer of nanomachines. At this point of nature comes to help. Molecular communications has evolved as an effective means of transporting and processing information from the replication of DNA to synthesizing complex molecules. Even a single cell in our body cannot survive without communication of fellow neighboring cells. They need the stay alive signal from other cells for sustained existence. Cells, organelles inside the cells, and even viruses can be considered as natural nanomachines. In molecular communications networks, encoded information is encoded on a molecular wave released by the transmitter nanomachine. This molecular wave propagates through the communication channel, typically a fluidic environment, via diffusion. The signal arriving at the receiver nanomachine has suffered a significant distortion exerted by the diffusion process. It is the task of the receiver nanomachine to decode and extract information from the received signal in a correct manner. The encoding and decoding processes may be in the form of concentration, number of molecules absorbed, or fluorescence intensity. Molecular communication is rather a new field, and most of the existing studies on micro-nanoscale molecular communications are mainly theoretical, generally assuming mathematical channel models to work on modulation problems. Here is a clear need for experimental verification of the proposed ideas that constitutes the aim of this study. In order to support theoretical simulation-based studies in molecular communication networks, microfluidics can be used to conduct molecular communication experiments. Microfluidics is a field that studies and manipulates a small amount of fluids in channels with length and diameters are in micrometer scale, provides an environment to conduct chemical, biological, and physical experiments in microscale. We have developed a microfluidic-based flow-assisted testbed using fluorescent polystyrene microbeads, which is widely used in biological tracing, in vivo imaging, and fluid mechanics, among other applications. Using the proposed testbed, our main aim is to create flow-assisted, microscale, and vessel-like environments to perform a comprehensive study on flow-assisted networks, to construct a building block for theoretical studies performed on molecular communications. The experimental approach includes 4 main steps, including laser ablation-based microfabrication, channel surface characterization by multiple analysis techniques, such as atomic force microscopy, stylus profiler, scanning electron microscopy, testbed construction and data collection, and at last, video processing. First step of microfabrication is the layout process, layout design of the desired microchannels for each chosen communication scenario. The design can be done with a CAD tool. We used AutoCAD. Next step is to fabricate. We used carbon dioxide laser ablation process to fabricate microchannels. This method is based on removal of certain quantity of material from a solid surface by irradiating the surface consecutively with a focused laser beam. Under continuous irradiation, the material reaches its transition temperature by the heat generated, and afterwards it evaporates. The method enables us to obtain variable surface waviness and depth due to different power level, skin speed, focal plane distance from surface used. Commercial 2 mm thick polymethyl methacrylate (PMMA) sheets were used during engraving experiments. The main goal of the microfabrication, this process, was to achieve reduced surface roughness, to make the channel a lot more like vessel-like structure for microchannels by heat exposure of a separate PMMA sheet at 80 centigrade degrees for 3 to 5 minutes prior to laser ablation process. Initially, optical microscope was used to analyze channel surfaces. Images were acquired at different focal lengths to achieve depth of field of microchannels. Then, ImageJ software was used to create 3D surface of microchannels by using those images. Additionally, we used a profiler to analyze surface roughness and channel depth. AFM commercial Bruker atomic force microscopy system also employed to compare the surface roughness values obtained through profiler. Aqueous solutions of fluorescence PS microbeads with 10 micrometer in size and blank aqueous media were introduced into microchannels from inlets according to communication scenario by using microsyringe pump. After injection, fluorescent beads were monitored and captured from observation window that was determined before at design step. Captured images is then analyzed by ImageJ. We used manual tracking plugin for ImageJ plugin, which provides XY coordinates in order to acquire XY coordinates of a single bead in a stacked images. The tracking plugin provides XY coordinates as well as velocity and distance achieved between two frames. For automated particle recognition and counting, a MATLAB-based image processing algorithm has been used. Surface roughness and depth of the channel greatly affects the flow type and behavior of the liquid. Finding optimum channel characteristics, depth and smoothness is significant for maintaining laminar flow that allows flowing without mixing of multiple layers of fluid next to each other. This feature of laminar flow provides the mimic cells and their environment. Therefore, depth and smoothness are critical characteristics for the microchannels. To find optimum depth and smoothness, initial tests were performed by changing the main parameters in carbon dioxide laser, its working power and its scanning speed. To determine the impact of the laser parameters on surface topography while keeping the scanning speed constant during engraving process, the cross-section of microchannel is determined by the intensity within the focused laser beam and the thermal diffusivity of the substrate. PMMA is a thermally stable material with low diffusivity, setting laser beam intensity as the main parameter for channel cross-section determination. Figure 4a shows the analysis of ablated channel profiler results, indicates a direct correlation between channel depth and laser power for different speed values. Here we see the symbols of different speed values. And at the lowest speed value, 25%, when we increase the laser power to 20%, from 20% to 100%, we increase channel depth at maximum level. And we see the minimum level in highest speed and lowest laser power. So we see a clear correlation. With increasing laser power and decreasing speed, beam can easily penetrate into substrate to engrave deeper microchannels. Hence, increased power and decreased speed results in increased depth of microchannel, and channel smoothness also correlates positively with increased laser power at lowest scanning speed, which is 25%. This is because the cross-section of the channel is directly tied to the shape of the intensity distribution and laser beam at constant focal distance, generating a top-hat structured beam shape at higher power levels. For molecular communications experiments, optimum conditions were chosen as 60% laser power, which is 18 watts, and 125 centimeters per second scan speed, which is 25%. Figure 4b shows reconstructed 3D topography surface mapping of microfluidic channel obtained with chosen value of speed and power. In figure 4c, profiler results indicate that at given parameters, channel depth is 470 micrometers and width is 506 micrometers. To reduce the roughness in microfluidic channels during engraving, microchannel fabrication procedure was repeated with a PMMA sheet exposed to heat treatment at 80 centigrade degrees before the laser engraving. Scanning electron microscope images presented in figure 4D and E. In figure E shows the preheated version of the PMMA sheet, which demonstrates that preheating approach clearly ameliorates the surface roughness while decreasing channel depth. This is due to the fact that preheating reduces the amount of dissolved gas in the polymer induced during production. During the laser ablation process, trapped gas can escape from the ablated area right before resolidification of the polymer, which gives the polymer the smoother look, with decreased depth and channel shape. We decided to continue the microfluidic flow experiment with chips obtained without preheating applied, the one shown in D. Later, microfluidic flow experiments were performed. A typical time sequential image of polystyrene microbeads retrieved from video recording beads are flowing in the microfluidic channels and recording made in bright field mode. The fusion of particles is ensured by laminar flow from injection point, Tx, to receiver point, Rx. The receiver point is not right. The receiver is actually this square, red square. Each bead is labeled in the recording like this. They turned red. And the reception was done by counting the particles as they move across the receiver zone. Here is the count, the yellow one. A microparticle-based molecular communication system using laser microengraved channels presented in this study. PS microbeads were released into microchannels through externally induced laminar flow. Each microbead, labeled as information carriers, was detected by microscopic observation. Using laser ablation-based microfabrication has simplified the process compared to other techniques for microfluidic chip production, such as photolithography, which is a slow and expensive technique that requires high maintenance cost. Most of the time, computerized simulations remain conceptual and build up theoretical knowledge, however not practical in real-life situations. This study is a bridge between computerized simulations and real-life applications. The PMMA microfluidic chip was used in order to mimic the blood vessel structure that is not fully absorbent nor reflective, so that the simulations are better aligned with practice. Therefore, the results of this study will allow us to develop channel models that will be applicable to real-life problems with the help of validation through testbeds. It is a proof-of-concept testbed that supports signal detection in microscale by using both bright field and fluorescence modes. Our preliminary results demonstrate that to build a chemical signal-based communication testbeds, multidisciplinary thinking and collaboration are crucial requirements. Thank you for listening.
Hello, I am Max and I will present our work regarding a comparative evaluation of a new sensor for spions in a molecular communication setting. In previous work, we presented a molecular communication testbed that uses superparamagnetic iron-oxide nanoparticles, short spions, as information carriers. The transmission channel is a tube with a constant flow of water. Data transmission is achieved by coding information as injected bursts of spions that are detected due to their magnetic properties. The challenge is to construct a receiver to detect the spions as they pass through the transmission channel. In the following, I will first describe the spions we used and the 3 sensor devices for comparison. After a short introduction of the testbed setup, I will detail the 3 evaluation procedures we performed and compare the results for the different sensors. Spions consist of a cloud of iron nanoparticles that is covered with a protective coating, in our case, lauric acid. As they were originally developed for medical applications, they are biocompatible. The particles we used were synthesized by the Section for Experimental Oncology and Nanomedicine of the University Hospital in Erlangen, and have a hydrodynamic radius of approximately 50 nanometers. Due to their superparamagnetic properties, causing a high susceptibility without remanence, they can be detected as inductance change of a coil, as described by the formula delta L equals the susceptibility chi times mu0, the coil surface A, the amount of windings N squared, divided by the length of the coil L. Therefore, the coil inductance changes proportionally to the core material's susceptibility. We compared 3 sensor devices. The first is the commercial susceptimeter from Bartington, which we used as a reference. It consists of 2 components, the actual susceptimeter MS3 and a range of adaptable sensors. In our case, we used the MS2G sensor shown on the left-hand side. The device is not optimal for our use case as it has various restrictions. Firstly, it has a fixed measurement aperture that unfortunately cannot be adapted to our testbed. This means that a transmission channel with a smaller diameter than the sensor coil causes a loss of sensitivity, Larger channel diameters cannot be used. Secondly, as it was not designed for continuous measurements, it has an insufficient sample rate of 10 samples per second and applies an internal measurement averaging. Acquiring the raw sensor data is not possible. We therefore constructed a new receiver for our testbed, the resonance bridge. It is adaptable to the testbed with custom-made coils and measures the change of inductance in the measurement coil as difference to a reference coil. The resonance bridge is operated at a fixed frequency of 10 MHz and is powered by a regular USB connector. The output is a DC signal proportional to the difference between the two coils and is recorded with an external acquisition module made by National Instruments. However, to achieve maximal sensitivity, a complex manual tuning procedure that requires an oscilloscope is necessary. The coils are made of bondable enameled wire with a width of 1 mm. Each coil has a length of approximately 2 cm and a width of 4 mm. They are attached to a printed circuit board fitted with SMA connectors so they can be easily swapped. The transmission channel is passed through the coil. The third device is an inductance sensor based on the LDC 1612 from Texas Instruments. The sensor measures the resonance frequency of a parallel circuit made up of a coil and a capacitor. As we observed some systematic errors due to environment conditions, we again used two coils, one for measurement of the transmission channel and a second coil as a reference. Furthermore, we used capacitors with a high rating for temperature stability to reduce effects of temperature drift. It was also fitted with SMA connectors to allow use of our custom-made coils. The measured resonance frequency is acquired digitally and transferred to a computer with a serial connection. As the accuracy depends on the measurement period, a trade-off between sensitivity and sample rate must be made. For comparison to the other devices, we chose a setting that allows for maximal sensitivity. We evaluated the capabilities of all three devices with the existing testbed. The transmission channel consists of tubing with an inner diameter of 1.52 mm. A constant background flow of water with a rate of 10 milliliters per minute is supplied by a peristaltic pump. Bursts of spions are injected at a Y connector with a second computer-controlled peristaltic pump. The Y connector is placed so that the injection occurs against the direction of flow, avoiding that particles are washed out of the injection channel. The spions travel through the transmission channel to the receiver and are detected with a coil. The distance from the point of injection to the receiver is 5 centimeters. Due to laminar flow, the bursts of spions are stretched out while they travel to the receiver inside the transmission channel. This causes intersymbol interference, significantly depending on injection volume and the distance between individual injections. The performance of the new inductance sensor was evaluated in comparison to the resonance bridge. The commercial susceptometer was used as a reference device. We investigated the device's sensitivity by determining the signal-to-noise ratio for various injection volumes, the overall capabilities for data transmission with a large random sequence, and the effect of the various sensor sample rates when using reduced symbol intervals for a transmission. First, we determined the bit error rate for each device with a sample transmission of 80 bits. This was repeated with 5 different injection volumes ranging from 7 to 84 microliters. The bits were detected with a simple algorithm in MATLAB. First, to get an equal effective sample rate of 10 samples per second for all 3 devices, a moving averaging filter was applied to the recorded signal. Then, a threshold was set at half of the maximal signal amplitude. The symbol intervals with a fixed length of 1 second were derived from the first rising edge. As the susceptimeter has a varying sample rate, the intervals had to be resynchronized again at every following rising edge. Finally, a bit was set to 1 if the threshold was met for at least 30% of the symbol interval. Here you can see a sample transmission with an injection volume of 84.1 microliters for each of the 3 sensor devices. The susceptimeter in red, the resonance bridge in blue, and the inductance sensor in green. The received signals were offset corrected to get a 0 baseline. In all cases, the individual bits of the 80-bit transmission sequence are easily detectable. Furthermore, the noise level of the inductance sensor in green is lower than that of the resonance bridge in blue. The low sample rate of the susceptimeter results in a representation of each peak with maximally 2 data points. Looking at the bit error rates for various injection volumes, we can observe that the bit error rate of the inductance sensor is consistently lower, and therefore better, than that of the resonance bridge. The results for the susceptimeter and the inductance sensor are similar. To evaluate the 3 sensor sensitivity, the average signal-to-noise ratio of multiple spion bursts were calculated for 5 different injection volumes. Here you can see the recorded signal for 3 individual pulses with different injection volumes. 84.1 microliters in green, 28 microliters in blue, and 14 microliters in red. A reduced injection volume results in a reduced signal amplitude at the receiver. Again, we can see that the inductance sensor's noise level is lower than that of the resonance bridge. Furthermore, the very low sample rate of the susceptimeter is clearly visible. Looking at the calculated SNR values for all 5 injection volumes, we can see that the signal-to-noise ratio of the inductance sensor is consistently better than the values of the resonance bridge. The susceptimeter exceeds both devices. To determine the influence of the various sensor sample rates, we repeated the transmission sequence with reduced symbol intervals, from previously 1 second per symbol down to 0.2 seconds per symbol. This was done with 2 injection volumes, 14 and 28 microliters, as larger injection volumes would cause too much intersymbol interference. For detection, we used the same algorithm as before. However, the recorded sensor data was now interpolated to 100 samples per second. For an injection volume of 28 microliters, we can see that the bit error rate increases as the symbol intervals decrease. Again, the bit error rate of the inductance sensor is consistently better than that of the resonance bridge. For a symbol interval of 0.2 seconds, the insufficient sample rate of the susceptimeter outweighs the better sensitivity, causing a similar result as for the other devices. At an injection volume of 14 microliters, the low sensitivity of the resonance bridge does not allow for confident bit detection anymore. The bit error rate lies around 40% for all interval lengths. The results of the inductance sensor and the susceptometer are similar to each other. In conclusion, the new inductance sensor has improved capabilities regarding both a higher sensitivity than the resonance bridge and a sufficient sample rate for transmission scenarios with small symbol intervals. Furthermore, the device is adaptable to the testbed with custom-made coils and is easy to operate as it does not require a complex tuning procedure. In future, we will work on optimizing the detector coil geometry, design a multi-channel receiver using more than one detector coil on the same transmission channel, and finally, improve on various testbed parameters, such as using a needle injection instead of a Y connector, and experiment with larger channel diameters. This work was supported by the Emerging Field Initiative of the FAU, the Städtler Stiftung, and the German Federal Ministry of Education and Research. Thank you very much for your attention.
Hello everyone, my name is Fatih Güleç. Today I will present our study proposed with Dr. Barış Atakan. This study is titled as Localization of a Passive Molecular Transmitter with a Sensor Network. The outline of my presentation is given as follows. First, I will introduce the concept of parameter estimation for macro-scale molecular communication. Then, I will present our novel experimental platform. Next, I will give the details about the system model and the sensor network-based clustered localization algorithm. Then, I will conclude after giving the numerical results. Macro-scale molecular communication can be used for applications such as environmental monitoring or detection of a virus propagating through air. In such applications, knowing or estimating the exact location of the source or the transmitter is important. The localization of the transmitter can be handled as a channel parameter estimation problem. In the literature of macro-scale molecular communication, there is only one localization algorithm, which is proposed for a long-range underwater scenario. However, this study is not validated by experimental data. Macro-scale molecular transmitters can be classified in two types as active transmitters and passive transmitters. Sprayers and pumps can be classified as active transmitters. Evaporating molecules can be classified as passive transmitters. In the literature of macro-scale molecular communication, passive transmitters are not studied experimentally. As the motivation of our study, we can say that there isn't any experimentally validated study for the localization of the transmitters in macro-scale molecular communication. And also, there isn't any experimental platform for macro-scale molecular communication with a passive transmitter. Since the propagation of a passive source is not driven by a force to direct the emission as the active sources, the usage of a sensor network instead of one sensor is reasonable for passive sources. Therefore, we propose a novel experimental platform consisting of a sensor network and a passive transmitter, and a novel algorithm for the localization of a passive molecular transmitter with a sensor network. Our experimental platform is deployed in a fume hood, which is a closed cabinet to conduct chemical experiments at controlled conditions. The sensor network consists of 24 MQ3 sensors and 2 Arduino Mega boards, which are connected to a computer. The passive transmitter is deployed at the midpoint of the sensor network. It consists of a petri dish which has evaporating ethanol molecules at room temperature. The pipette pump outside of the fume hood is only used to put the liquid ethanol into the petri dish before the transmission starts. During the experiments, we observed that there is always a wind in the medium, even in this small area. Therefore, we need to consider the wind velocity in our system model. The system model is the Gaussian plume model, which is widely used in the meteorology literature. From the conservation of mass, the first equation can be written, where S is the source term, C is the molecular concentration, and J is the mass flux. Here, J has two components. The first one is the diffusion component, and the second one is the advection component. The advection stems from the wind. Here, K shows the diagonal matrix of turbulent diffusivities, and U is the wind velocity. Hence, we can write the third equation as follows, which is known as the atmospheric diffusion equation in the literature. For an instantaneously emitting transmitter deployed at X sub t, Y sub t, Z sub t, the solution for a reflector ground is given as the Gaussian puff solution. The last exponential term represents the reflection of the plume from the ground. Here, the turbulent diffusivities are expressed in terms of dispersion parameters, which are sigma x, sigma y, and sigma z, as shown in the figure. The advantage of these dispersion parameters is that they can be determined empirically. For our scenario, the transmitter and the sensor network are deployed at z is equal to 0 plane. The concentration equation can be simplified as follows. Here, I and J show the row and column number of the sensor nodes respectively. You can see the deployment of the sensor nodes in the sensor network and the 4 directions that we used in this study in the figure. Based on the system model, now we can give the details about the sensor network-based clustered localization algorithm. This algorithm can be summarized by this block diagram. The first 4 blocks use the measured sensor voltages to calculate the detection time, molecular concentration, wind velocities, and the transmitted mass. All of these calculated values are taken as the input to the location estimator. The equation for the location estimator can easily be derived by using the system model equation. If we have 2 equations from the adjacent sensor nodes, we can solve the equations for x sub t and y sub t, which show the location of the transmitter. However, there are some unknown variables, such as the wind velocities, transmitted mass, and the molecular concentration, which I will explain next. In the algorithm, the first step is signal preprocessing and detection. The received signal for each sensor node is filtered with a moving average filter in order to mitigate the effects of fluctuations in the received signals. Then, the detection is made according to the threshold as given by the equation. For each sensor node, there is a different offset level which is shown by A sub O due to their characteristics. We also have a constant threshold amplitude which is shown by A sub T and is the same for each sensor node. By adding these two values, we obtain our threshold which is shown by gamma. The detection time and voltage value is determined according to this threshold. The next step is the sensitivity response of the sensor. The figure on the left side shows the sensitivity characteristic of the sensor in its datasheet in logarithmic scale. The next figure shows its curve-fit sensitivity function for alcohol in linear scale. This sensitivity function maps the concentration values to the normalized sensor resistance values. The figure on the right side shows the sensor measurement circuit. By using the parameters of the sensor measurement circuit and the sensitivity function, we derive a sensitivity response which is used to convert the measured sensor voltage value to the actual concentration value. After finding the detection time, we can estimate the wind velocities via the positions and detection times of the sensor nodes. We use cluster structure of the sensor network for the estimation. For example, for the x- direction, we can employ the sensor nodes in cluster 1. Or for the y+ direction, we can employ the sensor nodes in cluster 2. First, we calculate the wind velocity values for each node pair in each cluster. Then, we average these velocities in each cluster to find 4 velocity values for 4 directions. The last step for the location estimator is the transmitted mass calculation. For this, maximum velocity is found on each axis. Then, by using the magnitude of the velocity, the evaporation rate, which is shown by Q sub E, and whose formula is derived by an empirical study, can be calculated. The multiplication of the evaporation rate with surface area of the petri dish and emission time gives us the transmitted mass. If we turn back to the block diagram of the algorithm, we see that all of the inputs for the location estimator are found. Now, the location estimator can solve the equations for the location of the transmitter. As for the last step of the localization algorithm, we do not solve the equations for all of the sensor nodes. Instead, we utilize the cluster structure of the sensor network. Basically, the algorithm chooses the sensor nodes in 2 clusters with stronger velocities on x and y axis. Then, it solves the quadratic equations for the sensor node pairs of the chosen clusters. For example, if the wind is stronger on x+ and y- directions, we only solve the equations for the sensor node pairs in cluster 3 and cluster 4. In this figure, you can see the estimation results for each cluster. Since the wind velocity is stronger in the direction of cluster 1 and cluster 2, we have more results in these clusters. After eliminating the outliers, the estimation results for all of the clusters are shown together in the figure on the left side. Due to the stronger wind in the x- direction, the accuracy of cluster 1 is higher than the other clusters. Accordingly, the direction of the wind is visualized with the heat map of the average detection times for each sensor node in the figure on the right side. As we can observe in here, the wind is more likely to blow towards the left upper part of the sensor network. Lastly, we can see the estimation results in terms of the cluster error, which we derived as a performance metric. In order to calculate the cluster error, the Euclidean distance between the actual and estimated points are calculated for each node pair in the cluster. Then, these distances are averaged for all the measurements. This process is repeated for different threshold values. As we can see from the figure, the results are more accurate and stable for higher thresholds. However, as the threshold increases, the number of detected signals decrease. Therefore, this trade-off should be considered for the choice of the threshold amplitude. As for the conclusion, we can say that this study is a proof of concept practical application for macro-scale molecular communication channel parameter estimation with a passive source. We proposed a novel experimental platform using a sensor network and a passive source, and a novel algorithm for the localization of a passive source. Since our system model is originally derived for longer distances, it is expected to have more accurate results with our proposed algorithm in larger scales. Also, the usage of the sensor networks can pave the way to novel methods in macro-scale molecular communication by adapting techniques from the sensor network literature. Thank you for listening. I'm all ears for your questions.
Finally, I'd like to conclude the conference by announcing the best paper of BICT 2020. So the best paper of BICT 2020 goes to the following paper: Comparative Evaluation of a New Sensor for Superparamagnetic Iron-Oxide Nanoparticles in a Molecular Communication Setting. This paper is written by Max Batnick, Harold Unterweger, Christoph Alexov, Robert Schreiber, Maximilian Lubbock, George Fisher, and Jens Klickner. This paper addresses the new design, implementation, and evaluation of a molecular communication system. It's highly interdisciplinary and it best represents BICT 2020. Congratulations to the authors. So this is the end of the conference. This year, it is very unfortunate we cannot meet face-to-face, but we believe we can maintain the same quality of the conference, and we hope everybody enjoyed it. We'd like to thank again for everybody, including all paper authors, keynote speakers, reviewers, organizing committee members, and the EAI staff who supported the conference. We are now preparing for BICT 2021. We will announce the information as soon as it becomes ready. So please stay tuned and we will see you all next year at BICT 2021.